Okay, can you hear me? Hello?
Yes.
Okay, today we will
Yes.
focus what we missed out in this lesson, which is tooth structure, types of vitamins I remember, and we will go over deficiency diseases, and then the rest of the session, we will solve paper 6 together. Okay, so let's go directly to tooth structure. Okay. Now, this is a nice picture here of the different layers of the teeth. Fine. Now, the teeth is made up of a part that is above the gums. The gums is the red part that you see in the mouth. These are the gums. Here you have the gums. Now, the part above the gums that you see is called the crown. This crown is made up of the first layer. This layer is called the enamel. Okay? Enamel is the strongest tissue in the body, in case you did not know. And this enamel is made up of 99% calcium and phosphate salts. Okay? Enamel is very, very strong. Below the enamel, what do I have? Who knows? We took it last year. This layer.
Dentine?
Yes. The dentine or the dentine, they are both the same. See, if you notice here, it is written without an E, and here it is written with an E. So they're both correct. You can say it, or you can call it dentine or dentine. They're both correct. Now, the dentine or the dentine, it is a little bit weaker than the enamel. Okay, so it is made up of calcium also, but less calcium. And both the enamel and the dentine, if you notice, they don't contain nerve fibers and they do not contain blood vessels. All the blood is found in the central part of the teeth, which is called the what? The central part of the teeth, the most
Pulp cavity.
It's called the what? What did you say?
Pulp.
It's called the
The
pulp.
pulp cavity.
Pulp cavity. Very good. The pulp here, it is filled with blood vessels and filled with nerves. That is why when the cavity, when you don't brush your teeth and the decay reaches the pulp, this is when it becomes very, very painful. Okay? And the pulp, it provides the tooth with blood and nutrients, etc., etc. Now you have a layer here that holds the teeth to the gums. This layer is called the cementum, or is just cement is fine. So cementum or the cement, it holds the tooth to the gums. It's weaker, of course, than the enamel and the dentine, and it can also be carious if your oral hygiene is very, very bad and it reaches the cementum, your tooth will become loose and then it will fall out. So these are the different layers of the teeth. When you come to study it, it's very easy to draw it. Okay, I will draw a molar, for example, like this with 2 roots, because a molar has from 2 to 3 roots. And then this is one layer. And this is another layer. And I can do something like this, just to help you study. What's the first layer again?
Enamel.
Enamel, the strongest tissue layer in the body. Okay, it's stronger than your bones, by the way. Mainly calcium and phosphate salts. What's the second layer?
Dentine.
Yes, D, and this is weaker than the enamel. Both of them do not contain blood vessels. They might contain a little bit of nerves. That is why when the caries reaches the dentine or the dentin, you start to feel some pain. And then the central part here, it is called the what?
Bulb.
Pulp, yes. This pulp is filled with blood and nerves, and this is the most painful part. You have here the red part here that you see in your mouth. This is the what? The gum. And this tissue that is holding the tooth to the bone is called the what?
Cementum.
Yes, cementum or cement. Very easy, very, very easy. Now, you need to know the types of the teeth and how do they become carious, etc., etc. Let me use a better picture. Yes, this is really obvious. Here you have the 4 central teeth, those with a straight surface. They're called incisors, and we have 4 upper incisors and 4 lower incisors. They have a straight surface here, and in biology we call the
recording in progress.
Hello. This straight surface here is called chisel in shape. Chisel with an L, okay? So chisel means it has a straight surface like this. It's not pointed. It has only one root. So we're talking about normal cases, okay? So it is here flat from the top surface. In dentistry, this is called the occlusal surface. And it is chisel in shape. Chisel means it's flat like this. It has one root, and we use it to cut food. This is the use of the incisor, like, for example, biting an apple. And then after the 4 incisors, see, I have 1, I have 2, I have 3, I have 4 incisors. Now this 5th tooth here, the pointed one, is the canine. We have 2 canines upper and 2 canines in the lower jaw. So this is the canine. The canine looks like this. This is my canine. It's pointed. And this point in teeth is called the cusp. Okay, it's pointed. And the canines, of course, it differs in different organisms, like carnivores, like the lion and the tiger. They have a very pointed and very big canine. Okay, and the canine is used for tearing meat or tearing your food. And again, one big root. And by the way, in case you did not know, this is just general knowledge, the canine has the longest root. The root of the canine goes all the way to the base of your nose, in case you didn't know. Now, after the canine, you have 2 here, premolars here, 2 premolars. After the canine. These premolars, they are like smaller molars, okay? They look like this. Their top surface is containing 2 cusps, 2 pointed edges, and they are used for what?
Crushing and grinding?
Yes, crushing your food, to crush the food. Most of the food is eaten at the premolars and the molars. And the molars are just like the premolars. They are found at the back of the jaw, and they are just bigger than the premolars. They're just bigger in size, okay? And also, molars are more bigger in men than in females. If, let's say, for example, we have a corpse, we have a dead body, and that corpse is it's not in good shape, and we want to know if this horse is a male or a female. They can know from the molar, the size of the molar. When it is big and it has from 3 to 4 roots, it's big in size. It's usually this corpse is a male. I don't know why I'm mentioning that, but just general knowledge. So the molar is for crushing or the grinding of food, and it has cusps here, and it has from 2 to 4 roots. It depends, but usually they are 3 to 4 roots. Fine. So these are the different types of teeth. It's very easy, very, very easy. Now this is interesting. What is this? This is something called soft plaque. Okay, soft plaque. Now this happens when? Of course, this happens when you have very bad oral hygiene. You don't brush your teeth and you're eating. And this goes on for days and days and days. So the food, the remnants of food in your mouth, plus the saliva, okay, plus the bacteria forms a layer of soft plaque, just like you see here. Of course, this is very exaggerated, but if you don't brush your teeth for actually the whole day, by the end of the day, you find this thin layer of soft plaque. It smells very bad, and this is the first step in tooth decay. Okay, so this is soft plaque. This is someone did not brush their teeth for a very long time, maybe 2-3 days. And here's the plaque here, this soft layer of food and bacteria and saliva. It collects here and forms a yellow soft layer of plaque. It causes inflammation in the gums. This inflammation is called gingivitis, but this is too advanced. Now, this is here a hardened plaque, someone who does not brush his teeth at all, and this became hard. So he went to the dentist, and the dentist will clean and remove all the plaque, and now he has nice white teeth. Okay, this is the actual procedure of scaling, removal of the plaque. Good. Now, the important question, how does tooth decay start? You know what is tooth decay?
No, not really.
Tooth decay, the cavity, the susa.
When it wears out your enamel.
Yes,
and
yes,
reaches the bone
exactly.
part.
Yes, this, I want just to make sure that you understand what decay is. This is nice and healthy teeth here. Okay, these are nice and healthy white teeth, but sometimes they can go black and they can decay. Now, why would they go black? Because you're brushing your teeth or you're not brushing your teeth.
You're not brushing your teeth.
Brushing.
There's been bacteria actually.
Very good. You're not brushing your teeth, and you are a heavy smoker, for example. You eat a lot of sugary foods. You don't brush your teeth at all. Very bad oral hygiene. So you have food remnants in your mouth. You have food in your mouth. Food in your mouth that you don't wash away. And in your mouth, by the way, you have lots of bacteria. And this bacteria is anaerobic bacteria. Anaerobic bacteria means it will break down this food in the presence or the absence of oxygen.
Absence.
In the absence, very good. So what happens is that this food in your mouth will be broken down in the absence of oxygen. Who will break it down? The anaerobic bacteria. And this anaerobic bacteria will break down the food. Why does it break down the food? Because it is feeding on this food. It is using this food in respiration, in anaerobic respiration to provide energy. It's an organism and it feeds like any other organism. This food in your mouth will be broken down by the bacteria, and as it breaks it down, it will produce an acid known as lactic acid. This acid, yes, my dear.
Doesn't this happen in the muscles?
Yes, okay, but in the muscles and the enzymes in the muscles, here by bacteria, anaerobic bacteria, it's the same reaction. Glycogen or food is going to be broken down into lactic acid. Okay, sometimes you get confused and you tell me that the food will be broken down by anaerobic bacteria to give me carbon dioxide and alcohol, but then this is not by bacteria, this is by yeast. Okay, I don't want you to get confused. Anaerobic bacteria in your mouth breaks down the food in anaerobic respiration. Anaerobic respiration, we will study it in detail. For now, it is just respiration with no oxygen to give me what? Lactic acid. Now, this acid is strong enough and it can dissolve away the what?
Enamel ray.
Very good. It will dissolve the enamel, and if you leave it, it will dissolve the dentin, and the caries will go deeper and deeper and deeper until it becomes very painful. So this is exactly what happens. They can ask you a very easy question. How can you prevent tooth decay? Now, you don't even need to study a lesson to know how to prevent a tooth decay. What do you do so that you keep your teeth nice and healthy?
Brush your teeth regularly.
Yes, but they want several ways. So brushing your teeth is one way. Give me other ways.
Regular dentist visits.
Go to the dentist regularly, exactly. Brush your teeth, use dental floss, use fluoride toothpaste, avoid eating chocolate and candy. This is what we do with children. Yes. Yeah. Hello? Okay. If you have a question, just ask. We avoid sugar and candy to children because children's teeth are very weak. They are weaker than adults' teeth and they quickly become caries. So we reduce the frequency of the sugar. We said all that the food will collect inside the holes in the teeth and the bacteria will feed on the remaining food forming acids by anaerobic respiration. These acids will dissolve the enamel causing tooth decay. I like you to mention the name of the acid. It's lactic acid. And the caries will reach the dentin, making the tooth very painful. If the caries reaches the pulp, it will cause an abscess, okay? And the story can extend. Now, using a fluoride toothpaste, we will study why fluoride in specific, because fluoride makes your teeth harder, makes them resistant to decay. It makes your teeth harder to become decayed. That's why some countries, they add fluoride to drinking water. Fluoride makes teeth resistant to decay. Brushing removes the plaque and the food remains to prevent gum disease and decay. Make regular visits to the dentist, adding fluoride to drinking water. This is called fluoridation. Anything hard about that? Okay, this is, I think, general knowledge. I don't think you have any problems. Now, here is a question that actually asks you about the arguments for and against the addition of fluoride to public drinking water. This is a critical thinking question, but you will find it in your notes. I did add it to your notes. What does this mean? Outline the arguments for and against the addition of fluoride to public drinking water. Public drinking water means water that everyone is drinking. Now, they added fluoride to this public drinking water. And so we are all drinking water plus fluoride. Did they take your permission?
No.
No, they did not take your permission before adding the fluoride. So this is a disadvantage. I am against adding fluoride to drinking water because, where is this? No individual choice. Let's understand everything they said in the mark scheme. And also, some studies proved that fluoride can cause dark spots in children's teeth. So this is one against. And also some studies showed that the fluoride can cause some allergies. So let's read the against. The fluoride can cause allergies to some people. It can cause some side effects. It can make the water taste very bad. And the dosage, we don't know how much fluoride did they add to the water. This dosage is not controlled, okay? Also, you did not take my opinion before you added the fluoride, so no individual choice. And sometimes it will cause discoloration of teeth. This is the condition called fluorosis. This question came only once in the last 5 years. I never saw it except once, okay, or maybe twice. That's why I added it to the notes, okay, but still we have to be on the safe side. So we want to know that fluoride is a substance that is added to toothpaste and sometimes in drinking water to make our teeth stronger because it helps to strengthen our teeth. Okay, and when we add it to the water, it will be available to everyone because this is public water. So everyone will have access to this fluoride and it will be free. I will not pay extra money to add fluoride to my drinking water. These are the advantages. But the disadvantages, this might cause allergy to some people. It might give me a bad taste. You might add too much. You did not take my permission before you added the fluoride. And it might cause discoloration in the teeth of children. And this is called fluorosis. So is this question clear?
Yes.
Okay, anyone has any questions? So we only talked about the anatomy of the teeth and how does the dental decay happen, how to prevent it, and we talked about fluoride. I don't think you had any idea about fluoride. Okay, now why always the caries happens in the posterior teeth, in the molars? Always caries is common in the molars. Why?
Tape.
It's difficult
Excuse
to
me?
reach while
Have a large
brushing.
surface area.
Yes.
Has holes.
Yes, everything you said is correct. Again, this was a critical thinking question. You have to get used that sometimes in paper 4, they will get you critical thinking questions. In other words, questions that are out of the syllabus, but they want you to start thinking. And you know that this is a critical thinking question when they ask you suggest. When the question starts with suggest, then this is a critical thinking question. Okay? So when they ask you, suggest why molars are the most likely to decay than the remaining teeth, all of you gave me correct answers because the molars, this is your jaw. The molars are located here at the very back. So sometimes you cannot reach it. You do not clean properly. The molars, they're large. They have a large surface area. So they are exposed to the teeth. They have lots of holes. So maybe the food will be trapped in those holes. All these answers are correct. They have a larger surface area, so more food exposure, difficult to clean because they are positioned in the posterior position. They have holes or pits, okay, that trap food. So all these are reasons why the molars are usually the ones that become carious first, okay? I don't know why I put this picture, but I think I wanted to remind myself to tell you that we have 2 sets of teeth. We have teeth when we were children called deciduous teeth or milky teeth. Okay, they're smaller and they're weaker, and then they fall out, and then they will be replaced by adult teeth. How old are you? Year 10, how old are you?
15, 16.
Yeah, so you should have adult teeth. If you have any milky teeth, then something is wrong. Now, we will start here talking about deficiency diseases. No, we talked about that. Deficiency diseases. Let me see. What will happen or what type of disease will you suffer from if you don't have enough vitamin C?
Here.
You will have scurvy, okay? And the picture is obvious here. Scurvy or vitamin C is very important for healthy skin and healthy gums, healthy tissues and skin, for proper wound healing, okay? When you have a wound, any wound, the doctor will always prescribe vitamin C because vitamin C is very important for your skin, for healthy skin. That's why females put vitamin C on their face because it makes your skin nice and healthy, okay? If you don't have enough vitamin C, then you will suffer from weak and thin skin and your gums will swell just like this man, okay? This is scurvy. Of course, it's very painful. Your gums are inflamed and they're painful. This is a disease caused by the lack of vitamin C. Vitamin C in biology has another name. It's called ascorbic acid or citric acid. If you don't want to memorize them, it's fine. Vitamin C is present, of course, in lemon and oranges and citrus fruits. and the symptoms of scurvy, if you have this disease. By the way, we all have a percentage of scurvy because we don't ingest enough vitamin C, all of us, by the way. We have symptoms that include anemia, exhaustion, spontaneous bleeding, pain in the limbs, swelling gums. Now, of course, you will ask me about anemia. This happens in severe cases because when you have spontaneous bleeding, when your skin is very thin and you're bleeding for no reason, you lose blood. And when you lose lots of blood, then you will have the disease. What?
Anemia.
Anemia, okay? But this is in severe cases. This is when you have severe deficiency in vitamin C. So initially, the first thing you will notice when you have vitamin C deficiency is swelling gums, swelling and inflamed gums, and loss of teeth. Again, this is in severe cases. And gum ulcerations. Gum ulcerations means you will have abscess here in your gums. It's very bad, but no one reaches this state because it is very easy to treat. You can go and take vitamin C supplements and the condition will be resolved, okay? This picture is not related to vitamin C. This picture is related to the next disease here, which is rickets. This is another deficiency disease. Deficiency disease is a disease that results because you are deficient in one nutrient. Now, rickets is deficiency in which nutrient?
Vitamin D.
Vitamin
Vitamin D. Calcium.
Vitamin D first. Why is it needed?
To activate the calcium?
Yes,
And
vitamin
Open
D.
absorption of vitamin.
Yes, vitamin D, number one. It is stored in the fat under the skin. It is activated by sun exposure. That's why it's very important for children to play in the sun. Because when vitamin D is activated, it will absorb the calcium from your food. So vitamin D and calcium, they're friends. They work together to make your bones strong and your teeth strong. Okay? When your bones are weak, when you are a child and your bones are weak, no other picture? When your bones are weak, your bones will not be able to carry your weight and your legs will bend like this. And this is called rickets. And unfortunately, this disease has no cure. Okay? So the symptoms include bone pain, lack of bone growth, and soft, weak bones. Okay? Sometimes these soft bones will cause deformities, just like this picture. And for your information, so rickets is a disease for children. It affects children. But now as an adult, if I don't drink enough milk and I don't take enough calcium, I will also suffer from a disease that is similar to rickets, but it is not rickets. I will have soft bones. These bones are very weak and they easily break, which is also a very painful disease. Okay, so any questions about rickets? Any questions about rickets? No, good. Now, vitamin A. Vitamin A is also a very important vitamin for strong immunity and for healthy, what's this called? For a healthy vision. So if you don't have enough vitamin A in your diet, you will suffer another deficiency disease, which is poor immunity and poor vision. Okay, so poor immunity is another deficiency disease that results due to a deficiency in vitamin A. Vitamin A is very important for your immunity, okay, and for your healthy vision. Vitamin A, by the way, is found in carrots. That's why they tell you to eat lots of carrots, because carrots are good for your eyes. Another deficiency disease, constipation and colon cancer. What will cause constipation if you don't eat it?
Lack of
Fiber.
fibers.
Lack of
Fiber.
fiber.
Okay, I think you understand now that the fiber will add bulk to the food and will encourage peristalsis so that your digestive system is moving the whole time to push the food out. If this does not happen and all your food is soft, then you will suffer from constipation and, God forbid, colon cancer. Anemia is another deficiency disease. Here you are deficient in what?
Iron.
Iron. Okay? I think we mentioned this before, but I thought we should go over it. Now, this was also a question that came in one of the years. They asked you, suggest what can be the causes of malnutrition. Number one, what is malnutrition?
Lack of nutrients in your
Not
body.
having a balanced
Eating
diet.
not balanced diet.
Yes. Okay, very good. Malnutrition is either you are eating too much or too little. Malnutrition means you're not eating balanced. You're not eating the right type of food. You're overweight or you're underweight. So I'm eating too much food, I'm eating very little food, I might be of okay weight, but I have a deficiency disease because I'm not having enough vitamin C or vitamin D or fiber. All these are examples of malnutrition. Or too much fat, I'll have coronary heart disease, etc., etc. Famine, specifically famine. What is famine?
Starvation.
Very good. Famine is when a country has no food at all. No food, no water. They are in famine. It can happen through or due to severe poverty, natural disasters like a volcano or an earthquake or a tsunami. The soil is eroded for any reason, okay? War always causes famine, unfortunately, like what is going on at the very moment. You have very little land for very little people, for many people. The people are ignorant and they are not planting food. Overpopulation, unequal food distribution. You don't have to memorize all the 8 reasons, but 2 or 3 are enough. These are the causes of famine. Any questions? Any questions? Yes, ask.
Is rickets the same as osteomalacia?
No, rickets is in children. Osteomalacia is in adults. I just did not want to mention the name, but yes, thanks for reminding me. She's asking, is rickets the same as osteomalacia? They are both at different age groups. They are the same cause. We both, in both cases, there is poor or little vitamin D and calcium. In rickets, because the child is growing and his weight is increasing, the bones are too soft to carry his weight, so his bones will bend. And this case will be called rickets. But in adults, your bones are not forming. Your bones are formed. They are completely formed. But your bones become very soft. And you will have a disease called osteomalacia, which is soft bone in adults. Your bones are easily broken. Clear? Any questions? Okay, before I forget, I think I explained all this in class. The digestion is a long lesson. Do you want me to go over anything?
Miss,
Yes.
can you please go over the vitamin D? Like,
Okay.
what's has to do with calcium and stuff like that? 'Cause it's kind of confusing.
Okay, let me open the whiteboard and I'll go over it. It's very easy. Vitamin D, it's a fat-soluble vitamin. Let me open the whiteboard. Let me talk about the types of vitamins first. We want to mention the types of vitamins. You have 2 types of vitamins. You have water-soluble vitamins and you have fat-soluble vitamins. From the name, a water-soluble vitamin is soluble in what? So we are talking about the type of vitamins. You have water-soluble and you have fat-soluble. Water-soluble vitamins, from the name, these are vitamins that dissolve in water. The only example that you need to know is vitamin C. Any questions about vitamin C? Vitamin C is good for your skin, for your collagen, for healthy gums, etc., etc. Now, fat-soluble vitamins, these are vitamins that are found in fats. For example, vitamin D. This is the only example that you need to know. Vitamin D is stored in your fat. So if this is your hand and you have a layer of fat here, in this fat you have vitamin D. Vitamin D is found in fats. One of the functions of fats is the storage of fat-soluble vitamins. Now, when you are exposed to the sun, this vitamin D will become activated. Activated means it will start working. When it starts working, it will help absorb the calcium from your food. Don't you eat calcium? You will eat food containing calcium. This calcium needs to be absorbed. Who will absorb it? The activated vitamin C. So the activated vitamin D. So the vitamin D, its function when it is activated by sun exposure is to absorb the calcium in your food. When the calcium is absorbed, the calcium will be used for strong bones and teeth. So if you are drinking lots of milk, but you're not being exposed to the sun, the vitamin D will not be activated and you will not absorb the calcium. It's useless. They work together. The vitamin D has to be activated because when it is activated, it will absorb the calcium. The calcium is absorbed, it will be used in strong bones and teeth. Better now? Who was asking?
Yes, thank you, Miss.
Okay, now I completely finished every single point in this lesson, the core and the supplement. Anyone wants me to revise anything before I move on to paper 6?
Yes, Miss.
Yes.
Can you explain the villi again?
Yes, please.
Okay, fine. Let me use my PowerPoint so that I show you pictures. What exactly you don't understand about the villi? Here. I'll erase the board. Okay, I'm going to draw the inside of the small intestine. This is your small intestine. It's long and it's coiled. Now I'm going to look inside. The inside of the long intestine looks like this. This is the outer layer. The inner layer is folded. These folds are called the villi. And why are they folded?
Increase surface area.
Very good. These axes I am drawing here, this is food. I want to absorb this food. So when the villi is folded, this will increase the surface area so that the food is absorbed quickly. Now this is a single villi. I'm not okay with that. This is a single villi here. And I'm looking inside the villi. The villi is nice and folded, just like we drew here. And then this single villi, so this is one villi. This villi is lined with small cuboidal cells. It lines the villi. These small cuboidal cells, what are they called? Epithelial cells. These epithelial cells, they actually have two functions. They secrete enzymes, all the enzymes that we know about, the maltase and the amylase and the trypsin and the lipase, all the enzymes, because also some digestion takes place in the small intestine. Also, these epithelial cells, they have smaller extensions called what?
Microvilli.
And they have the same function as the villi, absorption. They increase the surface area for absorption. Okay, the food is absorbed. The food will be absorbed by diffusion, followed by
Active
transport.
active transport. And water is absorbed by
osmosis.
osmosis. Is this clear for everyone? Okay, now the food will be absorbed. We want to absorb it because we want to give it to the blood. How will they be carried to the blood? Just below this epithelial layer inside the villi, I have blood capillaries, the ones that I am drawing in red. The red small blood capillaries will absorb the glucose and the amino acids and the water and the vitamins and the minerals and all the small molecules. Now, some molecules are a little bit larger, and I'm talking in specific about the fatty acid and the glycerol. Who will absorb those large molecules?
Lactate or
Lymphatic.
Or the lymphatic vessel. The lactile is a little bit wider and will absorb the fatty acid and the glycerol. Well, they will all be absorbed regardless of who will absorb them, whether the capillary or the lactile, but in the end, they will all be taken to the blood. They will actually be carried by who?
Hepatic portal vein.
Very good. And they will go to the first organ, which is the
The liver.
Who is asking? Do you have any questions now? Is the picture clear?
Yes, thank you.
Very
Miss.
good. When you come to study it, draw it and label it, and this will help you and you will never forget anything. This is the villi. These are the epithelial cells, the epithelial layer, and they have smaller extensions called microvilli. And then you have the capillaries right here in red, and then a central lacteal. All the food will be absorbed by diffusion and active transport, water by osmosis. And the food will be taken in by the capillaries or the lacteals, the glucose, amino acids, and the small molecules by the capillaries, and the fatty acid and glycerol, because they're larger, they will be taken by the lacteals, and they will all be transported and go to the blood, to be transported to the liver to be filtered. Better?
Yes, I have a question.
Ask.
What about the lumen? Where is the lumen?
The lumen? Okay. I'll show you where the lumen, and this is the lumen, the inside of the small intestine.
Okay, so it's not inside the villi.
No, no, no. Inside the small intestine. It's the lumen, the inside of the small intestine. And sometimes some books call it the gut,
Yeah,
inside of the gut.
thank you.
You're welcome, my dear. These small questions, they're very small, but sometimes they confuse you. So ask me so that there is no confusion. Okay, let me move on to paper 6. I know that paper 6 is very long. We did solve some in class. We will solve some now, and we will never finish it all in class. So you have homework, and I gave you lots of time. I gave you homework until, I think, when is it due? 19th of November? I don't remember. But I gave you lots of time. Now, where is paper 6? 1 minute, let me open it. Who are you?
Miss, I have a question.
Okay, ask me.
Can you repeat the enzyme maltase or maltose? Because I get confused in this part.
Okay. You have maltose is not an enzyme. Maltase is an enzyme. Maltase. It ends with an ASE. Maltase is an enzyme. It breaks down the sugar maltose into what? Who will help me?
Maltose. Glucose. Glucose.
Glucose. Very good. Now, the enzymes that break down carbohydrates. We have 2 enzymes. We studied amylase and maltase. That's it. Amylase is released in 2 parts of the body. We have salivary amylase is released from where?
Mouth. Every
Yeah.
gland.
And we have pancreatic amylase. And by the way, we also have amylase released from the small intestine, but it's not called small intestine amylase. It's just amylase. So salivary amylase, what does it break down, my dear?
Source.
Into
Maltose.
maltose. So maltose is another type of sugar. It's just a disaccharide, okay? And the pancreatic amylase, it does the same thing. It breaks down starch to maltose. Now, maltose needs to be broken down further. Who will break down maltose?
Maltase.
Maltase. Maltase will break down maltose into what?
Glucose.
Glucose. Is the difference clear now? Any questions?
Miss, so in the test we just say that amylase breaks starch into glucose.
No. No. This is wrong. This was okay in the core. You can never say now that starch is broken down directly into glucose. It's a 2-step procedure. Starch is broken down to maltose. This is step 1. And then maltose is broken down by maltase to glucose. This is step 2. Okay, and they're done by 2 different enzymes. This is important. Okay, let me open paper 6, please. What do I want here? Paper 6, okay, digestion. What do I want here? If you have paper 6, please open it so we solve together.
Miss, I don't know what the homework is.
The homework is paper 2, 4, and 6, enzymes and human nutrition. This is the homework. Nice?
Okay.
When is the date?
Excuse me, where's the date? The date, I don't remember what date did I give you, but I gave you a long time. I gave you a very long time. But when, I don't remember. Like, it's not in a few days. It's a long time because check and tell me.
November 6th.
November 6th. Okay, you have a week. Enough? Enough or not enough? Let me open it. It doesn't want to open. I don't know why. Okay. This is cells and classification, digestion and enzymes. Okay. I think we solved a few in class. I want to start from this question here. We solved a lot of questions together in some classes, not all classes. So I want to start from this question. because this question was confusing some students. Okay, when we start working on paper 6 and read the experiment, we do the same steps. We will, what's this called? We will look at the experiment and we will decide what is the independent variable and what is the dependent variable. This is which page? This is 21 of 98. Okay, 1 minute. Okay. Now, let's read it together. Starch is an important food source that is digested by the enzyme amylase to form the reducing sugar maltose. So we were just saying that the starch plus water in the presence of the enzyme amylase will break down the starch into maltose. Some students investigated the effect of enzyme concentration on the rate of digestion of starch. So before I proceed, what is the independent variable? What did they study? The effect of what?
Enzyme concentration.
Okay, the moment you read the word effect, then what comes after that is the independent variable. So the effect of enzyme concentration, then they changed the concentration of the enzyme. What did they do? Step 1, 3 test tubes were labeled A, B, and C. So they have 1 and 2 and 3. Label A and label B, or test tube B and test tube C. Step 2, 5 centimeter cubed of starch solution was put in each of the test tubes. So they put starch here and starch here and starch here, and the same volume. Step 3, another 3 test tubes were labeled A1, B1, and C1. So another 3 test tubes. This is the best way to answer this question. Get a pencil, okay, not a pen, and draw step by step so that you imagine what is going on. This is the A1, B1, C1. So I am following step by step. Another 3 test tubes were labeled. Step 4, 1 centimeter cubed of 3% amylase solution was put in test tube A1. So what did they put in A1? They put amylase, the enzyme. 1 centimeter cubed of 3% amylase. 1 centimeter cubed is the volume. 3% amylase is the concentration. And 1 centimeter cubed of 2% amylase was put in test tube B1. So B1, they also added the enzyme amylase. And in C1, obviously, they also added amylase, but they added different concentrations of amylase. One time it was 3%, one time it was 2%, one time it was 1%. Do you understand what they're doing? Anyone is lost? All 6 test tubes, they're 6, see, they're 6, were placed in a water bath at 60 degrees for 3 minutes. Now let's stop here. Why were they placed all in the same water bath?
Reach equilibrium.
This is the answer to the question. If I had asked you, why did they leave it for 3 minutes? You will tell me to reach equilibrium. But why did I put it in the same water bath?
To keep the temperature constant.
Very good, because the temperature is one of the constant factors. Also, give me another constant factor. You have 2 other.
Equal volume.
Very good. The volume of the amylase. Both of them had 1 centimeter cubed of amylase, but of different concentration. All of them, or those 3 A, B, C, had the same volume of starch. So all these are constant factors. Step 6. A white tile was divided into 3 sections and labeled A, B, and C as shown in figure 1. So you will go and look at figure 1. Please, when you come to read a question like this that is long and detailed, read it with a lot of focus. Don't read it quickly. Read it with a lot of focus so that you don't miss anything. So they got a white tile and it is divided into 3 sections, A, B, and C, just like you see here. Iodine solution was dropped onto the tile to form 2 rows of 10 drops, approximately the same distance apart, in each of the sections A, B, and C. So they're just describing that they put iodine here. Iodine was dropped into the tile to form 2 rows. Okay, drop of iodine. That's it. Now, what else did they do? A glass rod was dipped into the starch solution in test tube A. So put in mind, don't forget, test tube A, only starch. We still did not mix anything. To remove some of the solution, the glass rod was then touched onto the surface of the first drop and then the second drop of iodine solution in row 1 on the section of the tile labeled A. The glass rod was rinsed and dried. So what did they do? They went to test tube A. This is test tube A here that contained only starch. And they took a drop out and they put 1 drop here and 1 drop here. So this first row actually contains only iodine. This is the part that a lot of you missed and you did not read. Okay? Step 9. Step 8 was repeated using the amylase solution in test tube A1 and the drops of iodine solution in row 2 on the section of the tile labeled A. So what did they do? Look what did they do. I want to show you what did they do here in this second picture. This is test tube A, test tube B, and test tube C. The first row is only starch. This is just starch. The first row is only starch. This first row. The second row is only iodine. I did not make this up. I figured this out because this is what they told me. And then when you come to read the remaining steps, in the remaining steps, they mixed the starch and the iodine, and then they took a drop out of the mixture. So this is the first drop when it was mixed. Do you all understand what happened? Who is confused?
Me.
Can you repeat?
Again, let's say we will talk about test tube A and A1. Fine, let's focus on A only. In A, I had starch, only starch. In test tube A1, I had the enzyme, amylase. They were separate. They were not together because once they will get mixed together, the reaction will start. I don't want the reaction to start. I want first to make them reach an equilibrium of 60 degrees. So I will put them in a water bath, these two separate test tubes, I'll put them in a water bath at 60 degrees for 3 minutes. So they reach equilibrium. And then what did I do? I took a drop out of the starch only. I still did not mix. I took a drop out of the starch only. I put it in the first row. And then I took a drop out of the amylase. I put it in the I don't know why I can hear. And then I mixed it. And then I took a drop out of this mixture and I put it in the third row. You understand now? What they did? These are just steps. Hello? Any questions? Can you hear me, everyone?
Yes.
Okay.
Yes.
Is it better now? Let's see what did they do. A timer was started and the amylase solution in test tube A1 was poured into test tube A. The mixture of the starch and the amylase in test tube A was stirred with a glass rod, and then some of the mixture was immediately removed using the glass rod. This glass rod was then touched onto the surface of the first drop and then the second drop of the iodine solution in row 3 on the section of the tile labeled A. The glass rod was rinsed and dried. This is what we said. This is what happened in row 3. So row 1, starch only. Row 2, iodine only. Row 3, this is when we started to mix. I hope you understand this because this is going to make a difference in the results. Going back, after 1 minute, the glass rod was used to remove some of the mixture from test tube A and touched onto the first drop and then the second drop of the iodine solution in row 4 on the section of the tile labeled A. The glass rod was rinsed and dried. So they waited for another minute and they took a drop out of the mixture and they put it in row 5 and row 6 and so on. This is exactly the same experiment we studied in class, but they made some differences. They said that we will take first a drop from the starch before we mix it and a drop from the enzyme before we mix, and then we mix and we take a drop in row 3. So row 3, this row, this is the start of the reaction. This is the difference that they made, and the spotting tile looks different. Step 11 was repeated for 6 more minutes. Step 8 to 12, so they did the same thing, was repeated for test tube B and B1 and C and C1. So they did that for the different enzyme concentration. Okay, I hope you understand what they did. Prepare a table to record the students' results. This table should include the concentration of the amylase solution and the time taken for all the starch to be digested for each amylase concentration. So what will be put in this table? What will be put on the left side of the table? What is the independent variable?
Independent variable.
Concentration.
The
or volume.
concentration. So the concentration will be put here. If I want to put the test tube, fine. We have 3 test tubes, A1, B1, and C1, or A, B, and C. It doesn't make a difference. And the 3 different concentrations and the unit. The concentration was percentage. A1, the concentration was 3. B1, the concentration was 2. C1, the concentration was 1%. I did not make it up. I went and I got it from the question. Now the time taken for the color or for the starch to be digested, the time was in minutes. This was the point of confusion. Put in mind that row 3 is the beginning of the mixture, not row 1 and not row 2. Row 1 and row 2 are used as control. They are used as reference because row 1 is only starch and row 2 is only amylase. Why did they put that? Because they will use these 2 rows as control, as a reference point. Starting row 3, this is when the mixture was done. This is when the reaction started. Do you all understand? Are you all with me?
Yes.
Okay.
Yes.
Why did a lot of you get confused and you asked me? Because you simply did not read the question. You rushed when you were reading the question. You told me, doctor, why is the time here in A? Why in A is it 2 minutes when it was 1, 2, 3, 4? No, you're wrong. You don't start from here. You don't start from here. You start from here, from the 3rd row. Because the 1st row, there was no reaction. The 1st row was only starch. The 2nd row, there was no reaction. It was only enzyme. So we actually start from the 3rd row. You just did not read properly. That's it. So let's look at the time needed for the starch in A to be digested. I'm going to start from the 3rd row. The 3rd row, they said they mixed it and they immediately took a drop. So this is time 0. So 1 minute, 2 minutes. Why did I stop here? Because the color here changed from blue-black to
Brown.
very good. This means that the starch has been digested or not?
Has been.
Yes. So test tube A or A1 took 2 minutes for the starch to be digested. And actually, the reason why, because A, the concentration of enzyme was the highest. It was 3%. Now let's count the time in B. Again, I'm going to count from the 3rd row. This is 1 minute. 1, 2, 3, 4, 5. Or 4. Is this an error or not an error?
Better.
Error, because you can see here that the drops have fused together. So the time in test tube B for the starch to be digested is 5 or 6. 1, 2, 3, 4, 5. Maybe you will tell me 6, maybe you will tell me 4. It's a range from 4 to 6 or from 4 to 5. Now let's look at test tube C. Test tube C was a concentration of 1%. Again, I'm going to start counting from row 3, not from row 1. This is 1 minute, another minute, a 3rd minute. And look at the 3rd minute. You have 1 drop that is blue-black and another drop that is orange-brown. Of course, this is an error. Something is wrong. So this is 1, 2, 3 minutes, 4, 5, 6. And at 6 minutes also, 1 is blue-black, 1 is yellow-brown. Is this experiment accurate?
No.
No,
No.
it's very poorly done. And then I have here the 7th minute. That is why the table will look like this. Where is the table? Where is the table? Here. Okay, this is the table. And for your information, you will come and tell me, I did not measure the time correctly. Will I lose marks? Yes. Yes, you will lose marks. Look at the mark scheme. Table drawn with 2 minimum columns and a line between heading and data. So there has to be a line and a table, at least 2 minimum columns. Appropriate column or row headings and appropriate units. So you have to have the units in the heading. That's why I keep on insisting, add the units, add the units. And then when you don't add the units, I deduct a mark. And then you become upset because this is what they will do. Okay? The units for the percentage concentration of the amylase enzyme for starch to be adjusted in minutes. So you have to have the units in the heading. You have to have the headings and the units. Another mark, 3 correct amylase concentrations, which is these, okay, recorded in any order. 3 correct timings recorded, okay. 6 correct timings recorded. You have this or this. 3 correct timings recorded for 2. That's why the time has to be recorded and the time has to be correct. So you have to read the question because you all did the same mistake. Those who were working on this question, you all counted the time from the first row. And you were asking me, why did I get different results from the mark scheme? Now you know. Because we count the time from row 3, not from row 1. Is it clear? Or I'm speaking to myself?
Yeah.
Good, thank you.
Yes.
Now let's move on. Explain why the students' results are not reliable. The common classic question, why are they not reliable? Let's go back and look at the results. These results were very messy.
Inaccurate.
Excuse me? What will you say?
It's inaccurate.
It's inaccurate. Yes, they are asking you, why is it inaccurate? Look at the results. It's very obvious what happened here.
The drops joined together?
Yes, the drops join together. So this is an inaccuracy. Okay, something else. Give me another inaccuracy.
They used glass rods instead of using a fixed volume.
Okay, you can say that, but this I think can be an answer how to improve, how to modify. Look at row C. Look at this row. See, I have 2 drops of 2 different colors. Don't you see that? I have one dark and one light, and I have the same case here. Don't you see that? It happened here, and it happened here. So this is an inaccuracy. So you can say that in row C, there were 2 drops of different color at 2 different timings. Fine. And they only did the experiment once. Did they repeat? Were there any repetitions? They did the concentration 1% once, the concentration 2% once, the concentration 3% once. There were no repetitions. So this is an error. You always need to repeat. Look at the answers. Drops for B at 3, 4, and 5, they have merged. We said that. And they only want one answer. So we will get the grade. Results for C have different end times. Results for C are different at 3 minutes here. Okay, this is 1, 1, 2, 3. So they are different. No repeats. So can you answer a question like that? Any questions before we move on? The starch was digested into simple reducing sugars. Describe how you could test the liquid in the test tubes to show that they contain reducing sugars. Classic questions, the same question that you took in the midterm exam. What will you do to make sure or to know if it contains reducing sugars? What's the test?
Benedict solution.
Benedict solution. Okay, this is 2 marks. You will just write Benedict and go home?
No heat at 80 degrees Celsius.
Very good. You will say that I'm going to add Benedict to the sample and I'm going to heat it. And it's very good to say that if the solution contains reducing sugar, what will be the color change?
Brick red.
Very good. From blue to brick red. Write the experiment because you are obliged to know it. State one variable that was kept constant in this investigation. We actually answered this question and we mentioned more than one variable. What is this variable that was kept constant?
Temperature.
Volume of
Volume of animals.
Yes, you are both correct. And how did we keep the temperature constant?
Water bath.
Statically
Very
controlled water
The
bath.
water bath was maintained at 60 degrees and we took the same volume for the starch and the same volume for the whatever. Explain why all the test tubes were left in the water bath for 3 minutes before the amylase was added to the starch. We also
For
answered.
thermal equilibrium.
Adjust temperature and get more accurate results.
Very good. So that they are all the same temperature, so that they all reach equilibrium. You don't believe me when I tell you that all the questions are the same format, but they just change the experiment. Explain why step 9 was carried out before mixing the amylase and starch together. I forgot what is step 9, so I'll have to go back and read step 9. What happened in step 9? Step 8 was repeated using the amylase solution in test tube A1 and the drops of iodine solution in row 2 on the section of the tile labeled A. Step 8, what happened in step 8? A glass rod was dipped into the starch solution. Step 8, what did they do? They took first a drop out of starch only and they put it in row 1, and a drop out of the amylase only without mixing and they put it in row 2. I actually mentioned that. Why did they do that?
So that the amylase won't react with the starch.
Very good. We don't want them to react yet, okay? And to act as a control, to act as a reference point. We took a drop from the starch before mixing, and we took a drop from the amylase before mixing, because we want them to act as a control or a reference point. We don't want them to get mixed, okay? When you understand, you realize how easy it is. Where am I? We want to finish this question at least. Where are we? I'm here. Identify 2 sources of error in step 10, 11, and 12. I will not go back. Can you think of any error? Always the error is the same. What's the error?
Drop size.
Excuse me?
Timing.
Timing, yes, the timing was not very accurate. But what is easier?
1 glass rod.
Drops of iodine.
Yes,
The drops was not the same size, or
you are all correct. All these are the same classic errors. Someone told me that the drops are not the same size. Of course, they're not the same size. How can you improve that? Here, how can you?
use a graduated pipette or a pipette.
Very good. I use a pipette, I use a burette, I use something more accurate. Another error.
Timing between the samples, no repeats.
Yes, all this is accepted. I did not do any repeats, so I will repeat at different concentrations. Color, please. Color is the easiest error. Any investigation that involves color change, color is an error. How do you improve it? How do you modify?
Colorimeter.
Colorimeter. Use a white background, colorimeter, repeat, okay? The timing, of course, putting 1 drop here and 1 drop here, the timing is not accurate. Maybe you will be fast, maybe you will be slow. So it is better to use 1 drop, not 2. It is better to do each experiment separately. All these are accepted. Excuse me, my dear, you're talking to me? Okay, now, in another experiment, okay? Some students made starch agar that contained 100 milligrams per centimeter cubed of starch. Now, I think I showed you what agar is. Agar is something like jelly, okay? So they made a cube of starch agar that contained 100 milligrams per centimeter cube of starch. The starch agar was stained using iodine and was then cut into blocks that measured 2 centimeters, 3 centimeters, and I don't know how to draw a cube, 2 and 3 and 0.5. Calculate the total mass of starch in each of the blocks of the starch sugar. Show you're working. Okay, this is 3 marks. Something like this can pop up at the exam. They made a block of starch, okay? And they want to know the total mass of starch in each of the blocks of the starch sugar. Again, in another experiment, some students made starch agar that contained 100 milligrams of starch per centimeter cubed. 100 milligrams per centimeter cube of starch. The starch agar was stained using iodine and then cut into blocks that measured 2 times 3 times 0.5. Calculate the total mass of starch in each of the blocks. You have 100, look here. How will you do it? I don't want to give you the answer. You tell me.
Calculate
the
volume.
of the agar.
Yes, the volume of the agar. How much is it? What will you do? Tell me step by step. What's the volume of this agar block? What's the volume? Tell me.
2 times 3 times
2 multiply 2 times 3 times 0.5.
0.5. Can you please calculate it? How much is that?
3.
3. Okay, this is 3 centimeter cubed of agar. How much starch is present in this block? This is not the answer. This is one step in the answer. I want to know how much starch is present in this block that has a volume of 3 centimeter cubed. How will I get it? It's here. The answer, look at this. Think.
300 milligram.
Milligram. How did you get it? Okay. How did you get it?
It said 100 milligram per centimeter cube.
Very good. 100 milligram of starch in 1 centimeter cubed. How many centimeter cubed of agar do you have? 3 in
3.
3 centimeter cubed. How much milligrams of starch? What will you do?
Times 100.
Multiply.
Times 100.
Exactly. 3 times 100. What will be the answer?
300.
300 milligrams. So I have 300 milligrams in this block of agar. There were 100 milligrams per centimeter cubed. When I have 3 centimeter cubed, how many grams of starch? They wanted to calculate the total mass of starch in each of the blocks. Do you understand how we got it? Any questions? 6 small beakers containing 20 centimeter cubed of 5% amylase were placed in water baths at different temperatures. So I have 6 small beakers, okay?
Miss, how do we know that we have 3 agars?
No, we don't have 3 agars, my dear. We have 1 block, but every block was cut 2 times 3 times 0.5. Read the question. Who said that we have 3 blocks? We have 1 block. Read the question. Read the question. Read the question carefully. The starch agar was stained using iodine. So I added iodine, and then it was cut into blocks. Every block measured 2 times 3 times 0.5. Every block had a certain dimension. We want to know how much starch is found in 1 block. You understand?
Oh, yes. Thank you.
Welcome. Good. Now, what did they do next? 6 small beakers containing 20 centimeter cubed of 5% amylase solution. So I have 6 beakers, and each one of them contains amylase, were placed in water baths at different temperatures. So what's the independent variable?
Temperature.
The temperature. Good. One of the blocks containing starch from DI, from here, so I put one of the blocks, was placed in each of the beakers. So the starch is going to be placed here into the Petri dishes. The agar cube that contains starch. And the time taken for all the starch to disappear was measured. The starch will disappear. Why? Because it is surrounded by amylase. The amylase will break down the starch in the block. So you understand what happened? You have here a block that contains starch and iodine. So it is blue-black in color. Let me use the color blue-black so you can imagine. I have a block here that has starch and iodine. It is blue-black. It will be surrounded by the amylase enzyme. What will the amylase do to the starch in the block? What will the amylase do to the starch in the block?
Break it down.
It will break it down. Very good. As it is broken down, how will the color change?
The color change
It
will
will remain.
blue black.
It was blue-black because this block is starch plus iodine. Starch and iodine will give me what color?
Blue-black.
Blue-black. So I started with a block that is blue-black. This block is surrounded by the enzyme amylase. The amylase will break the starch. It will eat the starch. When it eats the starch, the color will be changing from blue-black to what?
Brown.
Orange brown.
It will be the changing color of the iodine. Again, maybe I have something bigger. Look, I have a block. This block contains starch plus iodine. Starch plus iodine will give me what color?
Blue black.
Blue black. Okay, let me get the color blue black so that everyone understands. So I will start with a block that is blue black. I will put this block in a solution that contains the amylase enzyme. The amylase enzyme likes to eat the starch. I will digest the starch. So I will just give it enough time and all the starch will be broken down. The starch will be broken down. What will remain? The iodine. So this block with time, it will start blue black in color. With time, it will change from blue black to what?
Orange brown.
Exactly.
Brown.
Thank you so much. To yellow brown or orange brown, because it will go back to becoming iodine only. Clear? Okay, so we will measure the time needed for the color of the block to change from blue black to yellow brown. Let's go back to my question. Again, it's the same idea, but they are changing the method. Here they have a nice table in which they have the independent variable on the left side of the table, the different temperatures here, and the time taken for the starch to disappear, and the rate. I'm very happy that we're solving this question. Now, the rate, how is the rate ever measured? Always over time. Always a certain measurement over time. You took speed in physics? Didn't you take speed in physics? Even if you're not taking physics now, you took physics in year 8. Speed is always the distance over time. Any measurement over time is described in physics as the rate. So they want the rate of the reaction. And look at the units. In milligrams per second. Milligrams per second means what was divided over the time?
Mass.
Excellent. The mass, the milligram per second. They will ask you to find out the rate of the reaction when the temperature was 70. How will you do that?
Cross multiplication.
300 by 88.
Who said cross multiplication? Are we here finding out the concentration? I will not do any cross multiplication here. I'm finding out the rate. Whenever I am looking or calculating the rate, I have to divide a certain value, the dependent variable, my result, over time.
300 divided by 88.
Exactly. Excellent. How did I know what will I divide over time? Because it is here clearly mentioned in the units. It's milligrams per second. The mass over the time. The mass over the second. What's the time? 88 seconds. What is the mass of the block?
300.
Exactly. We already calculated the mass of the block, which was 300. So we will do 300 over 88. What's the answer?
3.4.
3.4. Very good. And you have to write it 3.4. If it was 3.56, you will not write 3.56. You will write 3.4. Why? Because you have to follow the same format as the remaining numbers. They are all to 1 decimal place. Then you have to write here the rate also to 1 decimal place. Clear? This is not a very easy question. Sometimes you can have an easy exam and sometimes you can have an exam with a tricky question. So you have to just be careful. Plot a graph. Any questions? Anyone has any questions, even if they are tiny questions? Ask. Plot a graph on the grid to show the effect of temperature on the rate of the reaction. Okay, so what will I plot on the x-axis?
Temperature.
Temperature, okay? So I'm going to plot the temperature on the x-axis. Please, I'm going to use the edge of the grid. I'm not going to make a graph inside the graph. I see very strange things. And I'm not going to divide the graph in half and use half the graph. I only do that when the values are negative. I don't have negative values. So I'm going to plot the temperature on the x-axis here, temperature. And don't forget the units, okay, in degrees Celsius. And what do I plot on the y-axis? What are my results? They said what on the
Rate.
rate of the action. Very good. And the rate, what is the unit? You tell me I don't know the unit. The unit is given, my dear. The unit is here, milligrams per second. So milligram per second. And again, I will explain how to plot a graph for the 10th time. I will not go look at the values yet. I will put here 1 mark and then 0. My pen is not working suddenly. Yeah, 0, okay? And please, you have to mark every block. Sometimes you do this, sometimes you do this. 1, 2, 3, 4, 5, and you say, for example, 1. Mark it. You have to make a mark so that the blocks are the same width. So I'm going to divide my graph into blocks. 1, 2, 3, 4, 5, block. 1, 2, 3, 4, 5, block. This is time consuming, but it has to be done. I will divide my graph. I'm going to quickly look at the graph and see how many blocks do I have. Wow, I have so many blocks. Okay, now we have to do it together. 1, 2, 3, 4, 5. If you have the paper 6, do it now. 1, 2, 3, 4, 5, and make a mark. Okay, it's very boring. 1, 2, 3, 4. And in the same case here, 1, 2, 3, 4, 5, etc. And then I have to distribute my values. How do I distribute my values? I have to go look at the maximum value. This is 70. I have to distribute my values so that my maximum value is close to the end of the graph. The 70 has to be here. It has to be here. It cannot be here. It cannot occupy half the graph or a little bit more than the half. No, you will lose a grade. The 70 has to be plotted at the end of the graph. Okay, let's go look at the maximum value for the rate of the reaction. It is 3.4. This is a decimal. Once you see that the rate of the reaction or the values are in decimals, it's easiest to start with 0.5, 1, 1.5, 2, and so on, until you reach the maximum value 3.4. And then you will plot...
The maximum value is 4.6.
4.6, 300 over 88 is 4.6?
No, before it, there's a
Thank you
4.
very much. I did not notice. Thank you. It's 4.6, okay? So why not reach 5.0? Again, 4.6 will be somewhere here. You occupied more than half the graph. Okay, so what's important is that the values have to be equally distributed and the maximum value has to be close to the end of the graph. And please label the 5 boxes, because if you put the numbers spontaneously, the boxes will not have the equal width. We don't want to lose a single mark for something that is very silly. I think you looked at your midterm exam and a lot of you told me, I did silly mistakes. I did silly mistakes. Those silly mistakes add up. When they add up 1 mark, 1 mark, 1 mark, then they add up and they can affect your grade. The final graph will look like this. Okay? See, why is this graph a correct graph? Because the x-axis is labeled and the units are there, so correct. And the y-axis is labeled with the units, correct. And then the maximum value is here, more than half the axis definitely, so correct. And the maximum value is 4.6 somewhere here, which is very close to the end, correct. And the plotting was done with x's, very good. And a ruler was used to join the center of the x's, correct. Always use the ruler. The ruler will make things very easy. So this graph will take a 4 out of a 4. Okay? Even scale, even scale. All the given plots are plotted accurately. Plotting is very important. Label with units, because you get upset when I keep on stressing on the units. Each line drawn with the ruler. And I keep on writing ruler, 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 okay? A smooth freehand curve through the points is accepted. That's why I did not deduct grades when you used your freehand, but a ruler is always better. Clear? Does this question end? Finally, it ended. Any questions?
Miss,
One question only, but one question included all the concepts of paper 6. Yes, what's your question?
I just want to ask what's a good grade for the, or like what's an A or A plus for the midterm exam out of 100?
Midterm?
Yes.
This question, if we would, because the midterm exam was a percentage of 100, so 90s is usually an A, high 80s is usually a B, okay? But I cannot tell you that this is definite because the curve is what really affects the grade, the curve in the final exam. What you should do is that you should do your best. That's it. And you should do
Okay.
your best in all three papers. If someone tells you paper 6 is only 20% and it is not important, then this someone is very wrong. Because in order to get an 8 or a 9, if this is what you're aiming for, you have to do well in all three, not only in paper 2 and 4. You also have to do very well in paper 6. Paper 4 is the direct reflection of how much you studied. That's why paper 4, you can solve it alone. You don't need anyone to help you. You don't need to wait for me. Paper 6 needs help. Even if you are an old student, it still needs help because it's tricky. Paper 2, I think paper 2 is very easy and you all finish it in class. Paper 4, we will solve together the critical thinking questions, the direct questions that ask you to define, to compare. You get it just as it is from the notes. You don't know the answer, then you forgot. You don't know the answer, then you did not study immediately. Paper 6 needs help. That's why we're solving paper 6 question by question. But this is not going to go on forever. I expect you to know how to solve it alone. Also, of course. Okay. I hope today was beneficial. And please work on paper 6 and paper 4. Now you can do all three papers. If you have any questions, I think now you know that you can send me a picture of the question and I'm going to explain the whole question for you. So you have no excuse. Fine?
Doctor,
Yes, my dear.
can you increase the due date?
Yes, I will. I don't think that 6 days are enough. It's 98 pages. We will also work in class, but I still think I'll give you more time. Okay. Bye-bye. Any questions before I go? We will meet again on Saturday. On Saturday, we will start reproduction. Good night. Bye-bye.
Bye.